We're talking about the new era for molecular breeding with cost-effective SNP genotyping solutions. Thank you.
Good evening. It's a real challenge to speak at the end, but I'm privileged to be the showstopper today because I believe after this there is no more oral presentation. I'll try to quickly take you to the journey of what we have been hearing since morning, that there is a scope to look into discovering genetic variations which can be screened on a large set of samples to validate those genetic variations and finally choose those few ones which it can be routinely used for a screening purpose, mostly called breeding programs. How do you choose the SNPs or how do you discover the SNPs? Today there are different models, different equipments, next generation sequencing, and there are tons of samples which you can run through whole genome sequencing program to discover those variations. This could be SNPs, this could be SSRs, this could be copy number variation, structural variations, depends on what technology you choose. What do you do with this large set of markers which you discover? You can run them through genome-wide association studies, linkage disequilibrium, multiple other sophisticated statistical programs to choose and refine those sets which can be finally used and can be linked to specific trait to define specific marker set to increase the probability of an yield or multiple other traits in your desired trait of interest. If I look at the plant genomes which have been sequenced so far, the first 50 till 2013 data I have, most of them are not covering more than 85% of the genome. And the N50, which is one of the figure which defines the quality of the genome assembly, are not very high. In this beautiful city of Hyderabad, these two monumental structure and the beautiful Hussain Sagar Lake everybody knows. And probably everybody knows this gentleman as well. Does anybody know this picture? You're not going to miss anything if you don't know her. Just like 30% of the genome sequencing or 15% of the genome sequencing which are still missed and you still have a draft sequencing to go ahead with. Do you know Hyderabad was originally known as what? Anybody from this area? Yes. This city was known as Bhagyanagar and this city was named of a beloved lovely lady called Bhagyamati who had been rather when she converted into Islam was named as Hyder Mahal and the city was named on her name as Hyderabad from Bhagyanagar to Hyderabad. These are a bit of information which you might not be very important, but probably in genome scale, you might miss certain information when you are not looking at those 15 to 30% of data. I am optimistic enough there might be some crucial information which might be very useful. There are technologies like Pacific Biosciences, which is extreme long read. This Limoji probably is giving you that view. That is today delivers somewhere around average 10 kilobases, where as high as 40 kilobases of read length, which has been achieved in laboratory and customers globally. What does this read length offer? This read length can actually give you a more holistic view of the genome, the portion of the genome which are much more complex, GC-rich, structural variations, and those structural variations, GC-rich regions, copy number variations might be useful in your science to pick up more valuable information. There is one study where this gentleman has run on Arabidopsis, Illumina sequencing has been used, and PacBio sequencing has been used, and you can see more than 250,000 new variations has been discovered just by increasing the assembly in a much more completion scenario. One of the examples where aluminum tolerance in maize has been discovered, which has been linked with one of the copy number variation, and this particular copy number variation with a very high read length of PacBio was elucidated, and that's how scientists have been able to understand why certain variety of maize has been able to grow better in a highly acidic environment, because this particular gene is reported to be one of the component of a transporter, cell transporter itself. One of the other example in switchgrass, where copy number variation has been also shown to be very, very important in the very biological processes itself. The other way, when you do not know the genome information, looking into the full-length transcripts might give you an opportunity to discover new variations. You do not know the genome, you do not have the ability to look for GBS based approach, but if you pick up the full length transcripts, it still gives you the opportunity to look at the variations which are there in the functional gene. And PacBio's technology with the highest read length will probably able to cover most of the transcripts which are coming from the genome itself. It's a fantastic approach to look at the genome variation discovery. I'm switching now to discovery mode to mid-scale genotyping, and Affymetrix doesn't demand any introduction probably. It has, to its own credit, has been serving the agricultural community with its solutions which have been ready-made catalog arrays. There are 22 catalog designs which are available today in multiple different plants and animal species, and from India we have been able to design arrays which are on rice, which are on cotton, and from ICRISAT itself, we are working with Dr. Bhaswar on chickpea, PGNP, and peanut. There are multiple publications which shows that these customers are having faith on us and they have been able to utilize this technology successfully in terms of getting quality publications as well. Looking at the demand of throughput, from a 96-well platform, Affymetrix has been able to launch a 384-well platform where you can increase your throughput for a routine screening with a lower number of marker density. So the two different distinct platforms where you are trying to validate a large set of variations which you have been discovering from whole genome sequencing or reduced representation library or any other means into a genome-wide association study to look into trait-specific markers, you would probably try to choose as many as markers as possible. In that scale, Affy supplies a 96 sample array plate where you can use as high as 675,000 markers at one go for each of the samples. Once you are done with this, you can look into the next scale with a defined set of markers which are trait-specific for a routine screening. And there are multiple such examples whereby ready-made arrays have been picked up. So Affymetrix covers starting from this region where you are looking into discovering marker specific to trait association and selecting out those markers and finally going into the screening phase. Minimum of 480 samples is required to commit, but you can run lower number of samples and we can really talk about how do I enable you to do that. These are the list of catalog arrays and the custom designed arrays which are used Axiom technology from Affymetrix, and this is only a representative list. There are multiple other things to add on. More than 26 crop species, vegetable and fruit, and more than 8 animals, and aquaculture also more than 8 species. So this is again another representation. The green ones are the plant one and the blue ones are the animal arrays which has been developed by Affymetrix in conjunction with the customers itself. One of the most interesting features over here, if you'd like to increase your throughput, at the same time minimize the cost, you can intelligently utilize the space which are there on the array. So you can use multiple different species and the variations which belongs to each of the specific species of interest in one particular array. So it allows you the possibility to share the real estate which is there in one array, multiple species, so that you can increase the throughput, you can lower down the cost, at the same time you can minimize the time as well. So when you talk about GBS as alternate approach, which is getting very popular, and we keep getting opportunity to discuss with multiple researchers, and to my experience, you have seen there are mixed bag of experiences globally. Some people are extremely happy because it's cheap, affordable, and you can run it faster. There are many outsourced service providers available. However, there are many questions coming in, and probably you have all gone through the understanding of a high throughput sequencing mapping process and what are the difficulties. There is one talk which you are exactly talking about what are the different challenges which comes through when you try to go through that process itself. And you are going to definitely miss certain data, because what we are trying to look for is to define this big animal by bits and pieces. It doesn't need any more explanation, but that's what exactly we are trying to believe, that the piece of information which you are going to pick up are the most valuable one to get what you are looking at. There are alternate technologies available in the market, and that does fantastic work and is done globally in terms of publications. However, Affymetrix also provides quality solutions in opposed to this in a little better way, where there is no batch-to-batch variations. So if you are planning your study on a larger scale, you are probably be more secure running on Affymetrix platform, because this technology which runs on beads, there are proven batch-to-batch variations available on that. Most of the plant species and the crops which are of your economic interest will be polyploid. And polyploidy is a bigger challenge when you try to look at the genomics data. And a couple of crops like wheat, strawberries, sugarcane, they're highly hexaploid, octaploid, or you are talking about cotton, which is tetraploid. For all these species, Affymetrix has created solutions which are more robust one to look into the data quality and differentiate the parameters which are being run by this technology. So you need a robust analysis software which will be able to segregate and polish the data which comes from this technology itself. So I'll quickly run through this. When you're looking at different kind of diploid versus allotetraploid and allohexaploid, allooctaploid, different kind of species orientation, you are going to get multiple different kind of peak variety. Your software solutions and the informatics has to be robust enough to differentiate quality solutions for the data which is being generated. And what this SNP polisher solution which comes from Affymetrix does, it categorizes in a normal scenario which you get 3 distinct clusters, but many a times you might miss out the 2 other one and only 1 cluster you might get. Sometimes there is something called OTVs or off-target variants, and they should be reproducibly coming up. You need to have a solution to look into those off-target variants. Sometimes the call rates will be so poor that there will be very clumsy data. These are the real scenarios, and these are not something only promises, because your everyday data will be coming like this. Affymetrix has listened to the customers, worked along with the customers to look into the solutions to differentiate and give you the quality structure when you are using the genomic solutions from them. And there are multiple customer feedbacks where they are saying they have tested Affymetrix softwares on livestock data, and that does a fantastic job to differentiate, give you the quality call rate. So quickly summarize what Axiom technology offers. There is no SNP dropout. If you have 6,000 markers, you are going to get 6,000 markers on your array. There is no marker dropout. There is no batch-to-batch variation, and you can choose 2 SNP as proximal as 10 batch pair different, and the automated call facility which comes from the inbuilt software. When you are doing this, at the end of the day, if you are lucky, you are probably going to get a very few SNPs which are probably in below 100 in number. We have another solution from Agena Biosciences which is called SICINome, and SICINome does a fantastic job by utilizing simple PCR and mass spectrometry based solutions in a very affordable and robust way. The distinct advantages are, because it doesn't use any fluorescence based data, you are not going to get any junk data. A mass which is the most accurate one, that's being measured over here. And the technology is relying on simple PCR. When you're using this is absolutely quantitative, because what we are going to get in a small number of markers, and you can actually differentiate different allele ratios by measuring the mass and the peak draft. The system is flexible. It do not need to look into a specific number of markers to a specific number of molecules. So the technology doesn't restrict you to a particular format of 24, 48, 96 or 384. You can choose as many SNPs as possible, as many samples as possible. That flexibility many a times will give you the most ease to take this technology forward. It has been globally used, and there are more than 2,000 publications on MassARRAY, and there are more than 300 instruments globally where people are using successfully in multiple applications, including Genomic. And as I was talking about, it works on a simple PCR-based technology to generate a simple primary extension product. What you measure are the 2 peaks, and the timeline from your data generation to the software analysis is less than 10 hours. Very comfortably, you can pick up markers of your choice as high as 40 to 50 at one go. Very low cost for genotype. Very easy to run because it doesn't need a very sophisticated instrumentation. It's just a simple MassARRAY based technology. Assay can be done by a single technician, and within 10 hours time you are getting results in your hand. I believe I'm pretty much on time, and I'm ready to take any questions.
Thanks very much. Other questions?
If you do not have a question, I have the last question to ask you. Do anybody recognize this figure? Okay, I'll move quicker. This is the picture of Balaji, commonly known as Visa Balaji. Those who has traveled from outside, I'd like to request, seek his blessings before you move for a seamless journey back home. Thank you so much.
Thank you very much. Please join me in thanking all the speakers for a very nice session, for the last session.
Okay, thank you very much once again, and then our sincere thanks to Eric and Shobha. So please join me in thanking them to organize this session nicely.